metabolic engineering in bacteria. So bacteria, they have been used as the source of host organism to express different type of proteins, hormones, and even the antibiotics. Initially, when the antibiotics they were discovered, like by Fleming in 1928, the antibiotic was penicillin, and this penicillin was used to cure different type of bacterial infections that were mainly caused by gram-positive bacteria. And here I can focus on the infections of staphylococci or streptococci, gram-positive bacteria. And the penicillin, it basically belongs to a group of antibiotics that are called as the beta-lactams. When this antibiotic was discovered, then its commercial production was started in 1940s to cure different type of microbial infections. But initially, the concentration of the penicillin that was produced in culture medium, it was very lower, and one can say that at that time the concentration was measured in microgram per liter. तो इससे आप ये आइडिया लगा सकते हैं कि इनिशियली एंटीबायोटिक्स की कंसंट्रेशन जो कि माइक्रोब्स प्रोड्यूस करते थे वह बहुत कम थी और इसको माइक्रोग्राम में मेजर किया जाता था, लेकिन ये जो क्वांटिटी थी ये सफिशिएंट नहीं थी to meet the demand. So for this reason, new strategies were required to produce the huge quantities of penicillin. For this purpose, the program was initiated that was based on the mutational analysis, that the strains of the microbes that produced antibiotics like the fungi, they were mutated by using different type of techniques, and later on the screening was performed that which mutated strains they have the ability to produce high quantities of penicillin. And after repeated steps, then scientists were able to isolate a mutant strain that has the ability to overproduce this antibiotic, that is penicillin, and the quantities it may be tenfold or even 100 grams per liter. So that was the huge quantity. But at that time, scientists they had no idea that what is the actual genetic or biochemical basis for the high production of the penicillin. Then with the passage of time, now we are aware that there are certain modifications in the metabolic pathways that ultimately governed by the genetic basis, they are involved in the overproduction of penicillin. And this strategy later on was used for the raw material of the artificial sweetener like the aspartame. And here the phenylalanine, it is the raw material for the production of this artificial sweetener, that is aspartame. Now, during the culture, it was noted that when the end product it is accumulated, then as a result of feedback inhibition mechanism, the concentration that was produced, it is arrested or stopped at a particular level. कि जब स्वीटनर की या एंड प्रोडक्ट की जो कि प्रोड्यूस होता है फिनाइल एलानीन अमाइनो एसिड से, उसकी एक ख़ास कंसंट्रेशन जो है वो लिक्विड मीडियम में प्रोड्यूस हो जाती है, तो उसकी वजह से क्या होता है कि उसकी फर्दर प्रोडक्शन जो है वो स्टॉप हो जाती है. तो इस प्रोसेस को मेनली फीडबैक इनहिबिशन कहते हैं कि जिसमें एंड प्रोडक्ट जो है वो अपने ही पाथवे के एंजाइम्स को इनहिबिट कर देता है कि फर्दर प्रोडक्ट जो है वो प्रोड्यूस ना हो. तो साइंटिस्ट ने ये मैनेज किया है कि जो फीडबैक इनहिबिशन का जो प्रोसेस है उसको मैनेज किया जाए और ज़्यादा जो एंड प्रोडक्ट है उसको हासिल किया जाए. So in the end we can say that now we have got the ability to manage the wild type microbial strains genetically and biochemically so that they can overproduce our desirable end products, that may be variety of antibiotics.